Hello and Assalamualaikum everyone. This is the video about biocatalysis and you are with me, Madam Wan Shaz. So, at the end of the lesson, you should be able to state the properties of enzyme, state 6 classes of enzymes, state the factors that affect the enzymatic reaction, define cofactors and define inhibitors. But first, what is an enzyme? Enzyme is a macromolecule that acts as a catalyst, a chemical agent that speeds up a reaction in the cell without being consumed by the reaction. This is taken from Campbell, 11th edition. How enzyme lowers the activation energy? So this is how it goes. With no enzyme, it's going to be a very high energy barrier, but then enzyme will lowers the energy barrier so that the reaction can take place easily. Next, let's have a look at the properties of enzymes. Number 1, most of enzymes are proteins. Number 2, most enzymes are globular protein and are coded by DNA. Number 3, enzyme acts as biological catalyst. Number 4, most enzymes are highly specific to their substrates. For example, enzyme sucrase will only react with sucrose. Number 5, enzymes have specific active site. Each enzyme reacts only with the substrate that is complementary to active site. The shape and chemical environment inside the active site permits a chemical reaction to proceed more easily. Number 6, enzyme will speed up the rate of chemical reaction. Number 7, each enzyme only catalyzes one type of chemical reaction. Number 8, enzyme will lower the activation energy needed for the reaction to take place. Number 9, enzyme is not altered or destroyed and can be used again after reaction. It remains unchanged at the end of the reaction. Number 11, enzyme is required in small quantity. Number 12, it denatures at extreme pH or high temperature. And the last one, the catalyzed reaction is reversible. The same enzyme can catalyze a reaction in both directions. Let's have a look at the classification of enzyme according to IUPNB. There are 6 classes that will be covered. Number 1, hydrolases. Hydrolases catalyze hydrolytic reaction that involve breakdown of substrate by addition of water. Sucrose plus water catalyzed by sucrase to form glucose plus fructose. Number 2, oxidoreductases. Oxidoreductases catalyze redox reaction. It involves transferring of hydrogen, oxygen or electron from one molecule to another. Example, ethanol plus NADH catalyzed by alcohol dehydrogenase will form ethanol plus NAD+. Number 3, lyases. Lyases catalyze certain reaction in which double bond is formed or broken without the addition of water. New functional group is added or removed by breaking or formation of double bond. Example, pyruvate catalyzed by pyruvate decarboxylase will form ethanol plus carbon dioxide. Number 4, transferases. Transferases catalyze the transfer of functional group from a donor molecule to an acceptor molecule. Example, glycogen plus inorganic phosphate catalyzed by glycogen phosphorylase to form glucose 1-phosphate. Number 5, isomerases. Isomerases catalyze isomerization. Example, glucose 6-phosphate with the help of phosphoglucoisomerase will form fructose 6-phosphate. Number 6, ligases. Ligases catalyze reaction in which two molecules joined and new covalent bond is formed. This reaction needs ATP as the energy source. ATP will be hydrolyzed. Example, amino acid plus tRNA catalyzed by amino acid tRNA synthetase will form amino acid tRNA complex. ATP at the same time has been converted into AMP plus 2 inorganic phosphate. Now, let's move to factors affecting the rate of enzymatic reaction. Number 1, substrate concentration. At low substrate concentration, there are many available active sites. Hence, the rate of enzyme reaction increases with increase in substrate concentration. But then, at high substrate concentration, saturation of active sites occur. The rate of reaction reaches the maximum reaction. Hence, increasing of substrate concentration has no effect on the rate of reaction. Factor number 2, temperature. Low temperature decrease the chances of substrate colliding with active sites, hence enzymes catalyzed reaction take place slowly. Increasing temperature, the kinetic energy of the enzyme substrate increase, hence the rate of reaction increases. At optimum temperature, temperature at which the reaction is at the maximum rate. At beyond optimum temperature, the rate of reaction decreases due to denaturation of enzyme. Number 3, pH. At low pH, ionic charge on the acidic and basic group of the peptide chain on the enzyme changes. Active site lost its shapes. Hence, substrate molecules no longer fit with the active site. The rate of enzyme reaction decreases. Increase in pH, rate of enzyme reaction increases until it reaches the optimum pH. Optimum pH, enzyme inactive state, maximum rate of reaction. But beyond optimum pH, the rate of enzyme reaction decreases due to denaturation of enzyme. Factor number 4, presence of cofactors. Cofactors are non-protein substances that are needed by enzymes to function efficiently. Cofactors. Cofactor is non-protein substances that is required by enzyme for catalytic activity. This is from Campbell 10th edition. Cofactor is stable at relatively high temperature. Enzyme cofactor complexes are known as holoenzymes. Enzymes that do not have cofactors are known as apoenzymes. Inhibitors. Inhibition, a reduction in the rate of an enzyme catalyzed reaction by substances called inhibitors. There are two types of inhibition, reversible inhibition and irreversible inhibition. Inhibitor, chemical substance that bind to an enzyme that inhibit the action of specific enzyme. So, any chemical substances that bind to an enzyme and decreasing the rate of enzyme catalyzed reaction or stopping its activity. So, for today's class, basically you have learned about the various properties of an enzyme that need to be understood. Number 2, there are 6 classes of enzymes according to IUPMB. Number 3, basically there are 4 factors that affect enzymatic activity, which are substrate concentration, pH, temperature, and presence of cofactor. And number 4, both cofactor and inhibitor affect the activity of enzyme in their respective ways. Thank you for watching this video. Bye-bye.